So in today's video, we're going to be looking at protein synthesis as we follow the exam guideline. Before we get into it, protein synthesis basically means the making of proteins. It is the process whereby proteins are manufactured in living cells. The manufacturing or making of proteins. So let's move on to the exam guideline. So here the exam guideline states that in protein synthesis we are going to be looking at the involvement of DNA and RNA in protein synthesis. Protein synthesis is divided into two stages or two phases, being transcription and translation. We are going to talk about the involvement of both DNA and RNA in the two processes. So with these two processes, you have to know them exactly how they are written in the exam guideline. This is how they're written in the exam guideline, these points and these points. And then the second point says that mRNA moves, And then afterwards, after looking at the entire process, we are going to look at a simple diagram to demonstrate transcription and translation. So let's start with transcription. So here we are under the first stage, transcription. The first point is that the double helix DNA unwinds. Like we can see over here, this is it unwinding. As it untwists, that is unwinding. And then the second step says that the double-stranded DNA unzips / weak hydrogen bonds break to form two separate strands. As you can see here, these two strands have unzipped as they unwinded. So they have broken apart into two separate strands. One strand is used as a template. You see this strand. It is the only one that has been used as a template, as in the mRNA is only forming on that one strand. To form mRNA, like I just explained, using free RNA nucleotides from the nucleoplasm. So in the nucleoplasm, let's say this is the nucleoplasm. There are free floating nucleotides that were used to form the mRNA. As you can see, these are nucleotides that are freely floating, coming onto the one strand that is acting as a template to form mRNA. And then the mRNA is complementary to the DNA. mRNA is complementary to the DNA, meaning that A will pair up with T, C will pair up with G, G will pair up with C, just like that. That's what complementary means. They follow base pairing, basically. mRNA now has the coded message for protein synthesis. Now this, we now call it a code or a message to then help with translation, the next phase that is coming. And then outside of translation, we now have outside of transcription, mRNA moves from the nucleus to the cytoplasm and attaches to the ribosome. So the phase where mRNA moves from the nucleus to the cytoplasm, we call it translocation, where mRNA transports itself outside of the nucleus to the cytoplasm through the nuclear pores. We call it translocation. Yes, we call it translocation. Let me try to find a picture to demonstrate what I mean. Here I am trying to demonstrate the translocation. You see over here, mature mRNA transport to cytoplasm through nuclear pore. We call this process translocation in between transcription and translation. So now we are moving on. Now we're moving on to the second phase, being translation. And then now looking at translation, each tRNA carries a specific amino acid. As you can see here, these are tRNA molecules, these ones. Due to the anticodon, they carry a specific amino acid. As you can see, this blue one is going to this tRNA because of its specific anticodon. tRNA brings amino acid to ribosome. So here, first step is tRNA carries a specific amino acid. We all understand that. When the anticodon on the tRNA matches the codon on the mRNA, then tRNA brings required amino acid to the ribosome. Yes. So now understanding that an anticodon codes for a specific protein, we also have to understand that this anticodon, these are the three nitrogenous bases together. Yes, when they are on tRNA, they are called anticodons. I'll explain it further later on. So when this anticodon matches the codon on the mRNA, then this tRNA will then go and deliver the protein to that specific codon on that specific segment of the mRNA. So that's basically what it means. And then we then say, like it said here, names and specific codons, anticodons and their amino acids are not to be memorized. It's always given in the data when they ask you questions. Amino acids become attached to each other by peptide bonds. You see these, when these are protein symbols, they are held together by peptide bonds. Yes, don't confuse the weak hydrogen bonds from the DNA molecule with these peptide bonds from the protein molecule. Two different things. And then second last point, amino acids become, last point, to form the required protein. So yeah, basically what I just explained. Peptide bonds are held together forming a protein that is required, basically. Yeah, that is basically it. So yeah, this is your 6-pointer answer for translation. Yeah, that's how it is. Now I'm going to further explain the codons and the anticodons. And we did use a simple diagram to illustrate translation and transcription. So yeah. So DNA, mRNA, tRNA, RNA. Here we call them a base triplet. When we come here, we call them codons. And then here we call them anti-codons. Yeah, this is not a hard thing to remember. Just know that in a DNA molecule, we call them base triplets. In an mRNA molecule, we call them codons. In a tRNA molecule, we call them anticodons. So this is when three nitrogenous bases are sitting directly next to each other like this. This will be something like this. So let me show you. Oh, let's just imagine that this is some sort of tRNA molecule. Not the best artist. Yo, wait. So, and then in here, it's adenine, uracil. It's too thick. And then in here, it's adenine, uracil, and guanine. In tRNA, we call these three base nitrogenous bases an anticodon, like I mentioned before. And then in an mRNA molecule, it's gonna be like this. Something like this. And then something like this, and then it's gonna be GUC. And then we come here. I want you to notice how in the DNA molecule, it is double-stranded. Unlike the other two RNA molecules, A, T, G, C, A, T. And I want you to understand that it is not by coincidence that here I'm not using and then I want you to understand that it is not by coincidence that I didn't use thymine in the mRNA and also in the tRNA. Remember that thymine is replaced by uracil in RNA molecules. So here you can see that in DNA we do have thymine over there. So when these nitrogenous bases are sitting in triplets, in DNA we call them base triplets, and if they are sitting in threes in mRNA we call them a codon. Sitting in threes in tRNA we call them an anticodon. So yeah, it's easy, it's not rocket science. So yeah, don't confuse the terms, they mean completely different things. So yeah. And then one last thing is that how to differentiate between transcription and translation. Which one comes first, which one comes second, because it does get a little bit tricky and you confuse them. This is just a little trick I used to use when I got confused during protein synthesis. So you see that both words have the word trans. So right after the word trans, comes the letter C, and here comes the letter L. Between these two letters, which one comes first in the alphabet? Obviously, C comes first. And L is second. So obviously, this one is the first one that comes, and this one is the last one. CL. Yes, I use the alphabet to manipulate my mnemonics and how to remember it. So if you do understand it this way, yes, you may use it. Yeah, translation and transcription. Yeah, that'll be it for today's video. Thank you very much.